traveling down the axon nerve fiber. As it passes by the voltage-gated membrane channels, they open and allow the calcium to flow into the axon. As the calcium concentration increases, the acetylcholine vesicles mobilize down and fuse with the axon terminal, where through excitosis they release the acetylcholine into the synaptic cleft. The acetylcholine then finds its way to the receptor ligand-gated membrane channels, which open and allow the sodium to flood in with its positive charge and depolarize the skeletal muscle. With the action potential is now ready to travel down the sarcolemma. The acetylcholine esterase in the synaptic cleft destroys acetylcholine, so another muscle action potential does not arise unless more acetylcholine is released from the motor neuron.
But that won't interfere with
The action potential is traveling down the sarcolemma membrane of the muscle skeletal cell. It's approaching an invagination of the sarcolemma, which is called the T-tubule. As it passes through the sarcoplasmic reticulum, the membrane channels will allow the calcium to release into the cytoplasm. So. The calcium will come over and bind with the troponin, which will then cause the troponin to pull the tropomyosin off of the active sites of the actin. When the active sites of the actin are exposed, the highly energized myosin head will leap over and cover the exposed sites of the actin. As soon as the myosin head covers the actin site, it will immediately go into a low energy state. And this is called the power stroke or the contraction. This is when the muscle has contracted. At this point, the ATP, the ADP, and the phosphorus will break free, and then the calcium concentration, as it starts to decrease by the calcium transport pumps, which are pulling the calcium back into the sarcoplasmic reticulum. They are held there by the calsequestrian, and when that happens and the calcium concentration has decreased, the muscle is now in a relaxed state. And ATP can come back over at any point and sit by the myosin head. At the point that the ATP breaks down into ADP and phosphorus, the head will be highly energized and it will be ready for the actin site once again to be exposed.
Here is a model of a fascicle, which is made up of bundles of myofibers. The myofibers are also made up of many myofibrils. Together, bundles of fascicles are what makes up a whole muscle cell. Within the muscle cell are many mitochondria, shown here by the orange structure. The mitochondria are responsible for holding lots of ATP, which is the energy needed for muscle contraction to occur.
This is a sarcomere. Sarcomere is a basic functioning unit of striated muscle. Sarcomere has thin and thick filaments. The thick filaments is the myosin, and the thin is the actin. Sarcomere has many binds and discs. The purple bands on the ends are the Z discs, the yellow bands are the thin filaments, and the pink bands are the myosin.